Okay, starting from now, 5, 4, 3, 2, 1.
Good afternoon. Thank you so much for joining Novogene's Global Research Live. My name is Lu Wang from Novogene EMEA. It is my great pleasure to welcome you to our Genomic Research Live session on behalf of the Novogene team. The COVID-19 pandemic has led to significant changes in the way people work and communicate with each other. Genomic Research Live is an online session facilitated by Novogene to provide a professional and borderless online sharing experience for life science scientists around the world. The session provides a symposium for global leaders in scientific, clinical, and business space to showcase their advances and progress in corresponding fields. This aligns strongly with Novogene's mission to advance the fields of genomics through sharing knowledge and cooperation. Today, it's our great pleasure to introduce Dr. Rui Xia, professor at South China Agricultural University. Dr. Xia obtained his PhD degree from Virginia Tech, USA, and had postdoc trainings at the University of Delaware and Donald Danforth Plant Science Center. His research focuses on genomics of horticultural plants, flower and fruit development, plant small RNAs, and bioinformatics. Today, Dr. Xia will speak about his work focusing on the domestication history of lychee and its flowering regulation decoded by full genome sequencing. Lychee is an exotic tropical fruit with distinct flavor. 稍后. Let's see how the genome of cultivar Rui Xia was assembled and phased, and how population genetics help to shed light on its domestication and fruit maturation among different cultivars. The webinar will last for 1 hour. If you have any questions during the talk, please leave your questions in the Q&A box. We will raise your questions to Dr. Xia after his presentation. If we don't have enough time to answer all of your questions, we will get back to you through email after the webinar. An on-demand version of the slides and recording will be provided after the webinar. We will send you a survey after the webinar. Please let us know how we did in the survey. Let's welcome Dr. Xia. Hope you enjoy.
All right. So should I start share my screen? Can you see my screen right now?
Hello, Dr. Xia. We see you from your video. Currently, we cannot see your screen.
Let me try. So right now it's okay. Can you see my screen right now?
Yeah, it's okay.
Okay, that's great. All right. Good afternoon, everyone. Thanks for Lu's introduction. I'm really honored to be here to have this chance to talk about our recent work on lychee genomics. It's a special time for all of us. We cannot meet in person, but we have this wonderful opportunity to talk about our research online. So in the next 1 hour, I will talk about our results on lychee genomics. Yeah, the topic is the origin and domestication of the lychee and the flowering regulation. So I'm Ray. I'm from South China Agricultural University. My major research focus on the horticulture plants, especially the flower and the fruit development. So for lychee, maybe you already know it's a special fruit, especially grows in the tropic and subtropical region. And the lychee is a species from the family, Sabindaceae family, which is, I think it's a big family. It has over 130 genera and also have around 2,000 accepted species. In this family, there are some famous fruit trees like the lychee and the longan and the rambutan. Maybe you already have tried those fruits. Yeah, if you come to the south part of China or in the subtropical region, you can easily find those fruits, especially in the summertime. And also in this family, there are some other economical crops like the soapberry, which we can use the fruits as a soap to wash our hair and hands. And also another plant like a yellow horn, which is the seed is rich in oil. We can use the seeds to extract the oil. The oil is very healthy. And this is a blue wine. It's a wine plant, and it can use as a herbal medicine. And another interesting crop in this family is the ackee. This is kind of the material. The fruit is the material for the national dish of Jamaica. Just like the national food, the national dish, you need to have the fruit as a ingredient. So this is an interesting family, but for this family, there are not many genomic studies for those plants. For the lychee, we can see, as I just mentioned, it is a subtropical plant. So it's distributed growth in this area, just like in the subtropical region, majorly. Just like from the 17 to the 26-degree latitude of the southern and the northern hemisphere. And we can see here, this region is maybe the Chinese Taiwan, China, and the Vietnam, and maybe Thailand, India, Pakistan, and Mexico, Brazil, and also Australia. Some part of Australia also have the lychee growth. So for the production, we can see just like this from the pie chart, we can see just like the China has the largest production. It accounts for maybe 70% of overall production worldwide. And follow the China is maybe the Chinese Taiwan, or not the Chinese Taiwan, it's India. India is the second one, and the Pakistan is another, the Vietnam is another one. Within China, we can see just according to for those provinces, the Guangdong province, which is here, where I'm right now in this province. My university are located in this province, and it has the largest production of lychee in China. It accounts for around 60% of overall production. So that's why so we are saying that lychee is a really important crop in my region. Lychee is the second most important subtropical or tropical fruit worldwide, and it has a long history, cultivation history of over 2,000 years. So in ancient times, those lychee we harvested in the south will be transported to the north, where the king lives, just over thousands of miles for the transportation. Yeah. And many of you, I guess many of you have already maybe know how the fruit tastes. Yeah, this is the fruit, and we can see it has a colorful skin, and the meat, we call it area. That's the flesh we eat, and the seed inside, and this is a tree look like. The tree is pruned, just like not the normal tree. The branches have been cut off. Yeah, for the lychee industry, as I just said, it's important in my area, but there are still some really bigger problems. One is just like the harvest time is short. Yeah, we can only find the fresh fruit within 3 months, the May, June, and July. Yeah, you can only have fresh lychee at this period of time. After that or before that, you cannot find it. So this gives a big problem to the industry, since all the fruits comes to the market at almost the same time, give a lot of pressure to the fresh market. Then the price will drop, the profit of farmers will become low. So that's I think is the biggest problem. And also the fruit, you may know that the fruit is not good for post-harvest storage. It cannot store it in the refrigerator for a long time, not like apple or citrus, right? And I think one reason give a problem of the short harvest time is that we don't have the kind of the extremely early or late cultivars, just like all the cultivars have similar harvest time. So for the breeding, just like the lychee, we still use a very traditional method for the breeding, like the hybrid, just like crossing, and also maybe incidental selections from natural mutations. But for other crops, we already use maybe the third or the fourth generation technologies for the breeding. So in this part, our lychee has a long way to go. So to solve these problems, I think the number one thing we have to do is to decode the genome sequence, which will be helpful for our understanding of the kind of the mechanism, how to solve those problems. So maybe 10 years ago, our department, just like at the SCAU, the South China Agricultural University, we start this genome project. We select one of the widely cultivated variety for the genome sequence. It's a Lychee. Yeah, this is a famous cultivar in our country and in our culture. It means the smile of the princess. Yeah, it's famous in our culture. So after taking advantage of all kinds of sequencing technologies like PacBio, the Illumina and HiC, and even 10 genomics, we got a really good quality reference genome. Just like the heterozygous rate is around 2 and 27%, and the chromosome is 15, and the genome size is around 500 megabase. So, and we got around 30,000 genes. The completeness looks quite good. Yeah, see, it's around 95% for the genes, and for the genome is around 96% completeness. So overall, this is, I think, is maybe the best quality genome in the Cephalaceae family. So here we have 3 in the Cephalaceae family. We already have 3 genomes: the yellow horn, the longan, and the lychee. Based on the tree, use the single copy genes, we construct a tree like this. We can see at around maybe 28, meaning 68 million years ago, the Cephalaceae species splitted from the citrus. And after that, there's almost no kind of whole genome duplication events. Yeah, here we can see it's the gamma triplication event, which is common for all, I think it's angiosperms. So there's no, based on our data, there's no WGD events in Lychee. How? Okay, so after we got a very, just like a good quality reference genome, yeah, we used our data to do another thing, which is quite interesting. Since at the early years, we typically do a genome, we combine those two homologous genomes into a reference genome. So the reference genome is always a merged one, just a chimeric one. It's not a real one. So, but because the high heterozygous rate of our physical cell Lychee, so we try to separate the haplotype genomes. So just like this, because of the high heterozygous rate, it enabled us to separate the two sets of haplotype genomes. Yeah, for those haplotype genomes, I think the quality looks quite good. For the gene numbers and the length of the chromosomes, although the size dropped a little bit, but it's still of a quite good quality. And for the completeness evaluated by the BUSCO, we can see it's still quite good. It's around 90% for the genome, and for the proteins, it's around 85%. I think for a haplotype genome, it's of relatively good quality. Yeah, that's the genome part. Yeah, we sequenced the physical cell genome. We got a high quality reference genome. We also got the haplotype genomes. After that, we want to just as maybe in my title already told, we want to know the origin and domestication history of lychee, right? So we connected many different varieties of lychee over my country, China, and also some from the Vietnam. We got around 72 accessions, and include 38 wild individuals and 34 cultivars. And here, those red dots indicate the wild lychee. Yeah, there are 6 locations has the wild lychee, and those 2 big ones indicate the 2 largest populations of wild lychee. Just one is in the Yunnan province, the other is in the Hainan province. And from here we can see the fruit looks quite different. The size of the fruit and also the skin color and the size of the seed all looks different. And shows a lot of diversities. After we got those samples, we did the resequencing. We cut the SNPs and did the cluster analysis, and this is the tree we got. From this tree, we can easily classify all those accessions into 3 groups. One is on the left side, we can see it's, we call it as the extremely early maturing cultivars, EEMC, and also include the Yunnan wild lychees. And here is the Vietnam wild lychees. And also on the right, we can see there are some later ones, later maturing cultivars, and also include the Hainan wild lychees. In the middle, here in the middle, there are a few of them, just like there are early maturing, just like the maturing time is in the middle of the extremely early and the later ones. After we got this clustering results, we did an interesting analysis. We as in the first part, I said we separated the physical lychee into kind of the genome into 2 haplotypes. So we mapped all the resequencing data, just like the second generation resequencing data, to the haplotype genomes, and calculated the percentage of the coverage. From here, we can see the 2 colors indicate 2 different haplotype. And the dotted line here is the 75%, the middle one is the 50%, and the lower one is the 25%. So from this plot, we can see just like for this part, this population is the late maturing one and the highland wild ones. We can see over 64% of the coverage constitute the allied to the haplotype 1, the yellow one, right? Just like around 70% of the rates allied to this one, just like the yellow one. But for the extreme early one and the Yunnan wild, it majorly allied to the second haplotype, the blue one. It just like more than 60% of them allied to the second one. So this is a piece of interesting results. And for the middle one, for the extremely early, for the early maturing ones, EMC, we can see they are quite half to half. So from this result, it gives us a kind of indication. So the yellow one is a haplotype from highland. I think it comes from the highland wild lychee, and it constitute maybe involved to the late maturing cultivars. But for the other haplotype, it comes from the Yunnan wild, and it's domesticated into the extremely maturing cultivars. And the crossing of those 2 materials due to the early maturing cultivar include the Xiao. So based take all those data together, we proposed a hypothesis for the origin and the domestication of lychee. One is the first one is the one original center, just like I think the original center is the Yunnan. Just like the wild lychee in Yunnan will spread to Guangxi and Guangdong, and eventually comes to Hainan through the Xinjiang River, and also maybe the people transfer, right? So just like and after the geographic movement, just like the strait formation, the Hainan population become isolated, become separated. So that's why we got 2 major, just like wild population of lychee. One is Yunnan and the other one is in Hainan. And for those 2 major populations, they have the domestication separated. The Yunnan wild is domesticated into the extremely early ones, and the Hainan wild domesticated to the later maturing cultivars, and the cross between them give rise to the early maturing ones, include the Xiao. So this is the whole story for the origin and the domestication. For the hypothesis, there is still a minor question we need answers. We know the crossing of LMC and the EEMC give to the EMC, but which one is the father side and which one is the mother side? To solve this minor question, we used the resequencing data. We assembled the chloroplast genome and did the cluster analysis. We can see here from the Xiao clustered together with the Hainan wild lychee. So we all know the chloroplast genome mainly come from the mother side. So we are saying here, just like the the later maturing cultivars is the mother side, and the EEMC ones is the father side. They cross together, give rise to the phase shell, the EMC one. So this is an interesting kind of small piece of information. Yeah, in our study, since we got as I mentioned, we got a really good haplotype genomes, so we are able to do our genome-wide kind of analysis for the differentially expressed alleles. Yeah, from this plot we can see we used all the libraries we have around the 40. We can see for the number of DEAs, differentially expressed alleles, we can see the number increased, and once the library number reached to around 40, the number of DEAs reached the plateau, right? So we got around 13,000 DEAs, and for those DEAs, we checked the Ka-Ks ratio. Just like we found this DEAs has a much lower SI, significantly lower Ka-Ks ratio, indicated those DEAs have a greater, just like a bigger purifying selection pressure. And we also checked the SNP density for those DEAs and the EEAs. EEA, just like equally expressed alleles. We can see here, for those genes, the SNP density is much higher in the promoter region, which makes sense, since we need to have differential expression for those alleles, the SNP, the promoter region have SNPs will cause maybe the different rate of transcription, right? And the exons has a much lower, just like SNP density. Yeah, to keep the kind of the protein sequence unchanged. So yeah, and at this part we also checked the exonic SNPs, those SNPs in the exons, see if they will change the protein sequence or not. Indeed, we found the majority of those SNPs in the exons will change the sequence, and the changes from transition is much more frequent than the just like transversions. So those are the kind of based on the dry data analysis, we found some interesting information from the genome of Lychee. The second major part of my story will focus on the just like a flowering time regulation. Just we are trying to use the genome, the data from our resequencing data to dissect the mechanism, to understand more on the flowering. As I briefly introduced in the beginning, the biggest problem in the lychee industry is the short fruit harvest time. The maturation time is short, less than 3 months, right? It's mainly in the May, June, July, and a lot of fruit will get into the market, give a lot of pressure to the fresh market. That will cause lower price and lower profit for the farmers. So how to extend the maturation time to ensure a longer supply of fresh lychee is essential for the lychee industry. But how does the harvest time and the maturation time controlled in lychee? Normally there are two factors. One is the flowering time, just like what time the plant will flower. And the second one is the number of days from flowering to fruit harvest. But for most cultivar, for most varieties of lychee, the number of days from flowering to fruit harvest is almost the same. So the flowering time is the number one and determining factor for the maturation time. So that's why here we focus on the decoding of the flowering time control in the second part of our story. For the flowering control, there are many studies in model plants. So we use all the kind of flowering related genes to do eye search in the Lychee genome, identified all the flowering related genes, around 500 genes. And for those genes, we did a wide profiling of all those genes to try to find some interesting part. Indeed, we found a few of them. Here, I will give you 3 examples. The first one is the SVP gene cluster. SVP gene is important for flowering. I will tell you a little bit later. Just like here, from the syntenic block analysis, we can see here. Just like this is a yellow horn, and this is longan and the lychee. We can see those red ones, the red line connect those SVP genes. We can see here in longan, there are 8 genes, SVP genes, and in lychee, there are 8 genes as well. But in yellow horn, there are only 1 gene. So it seems like the SVP genes has a big expansion, duplication in longan and the lychee genome. So for the SVP genes, it is a matchbox genes. It is kind of one of the famous genes in the flowering time control. It controls flowering by negatively regulating the expression of FT. It will repress the FT expression binding to the promoter of the FT genes. And for the expression, we can see here, all those duplicated SVP genes expressed heavily in the leaf and the leaf bud and the flora primordial. Which this expression pattern is highly similar to the expression of SVP gene in Arabidopsis. This indicates the function should be similar, just like it also has some function in the flowering time control, we believe. And also there is another piece of information is interesting and related to this cluster. Indeed, in the peach genome, we can see there is also a cluster of 6 SVP genes in the peach genome. The deletion or part deletion of 4 of the SVP genes will give rise to a mutation. For the mutation, the plant will grow without stop. Since normally in winter season, the peach will stop, right? But for this mutant, it will not stop. It will keep growing, have flower, and even you have a low temperature. So this gives us a strong, just like evidence to believe our SVP cluster will have function in the flowering time control. But right now we don't have solid data. We cannot do we have to connect more data to support this. Another case I'm going to talk about is the VRN1 gene cluster. Here we can see, also for the syntenic, we can see here those blue ones, light blue ones are all SVP genes, and this red one is the closest homologous to the Arabidopsis one. So we colored it as red. So here we can see from this syntenic plot, we can see there are 24 VRN1-like genes in this region in Lychee, but 17 in longan, and as many as 26 in the yellow horn, and only 7 in orange. So what is VRN1 genes? VRN1 genes was first identified in Arabidopsis as well. It is important in the volatilization. It will it is associated with the FLC. It will inhibit the expression of FLC and then control the flower of plants, just like it will respond to the temperature and will repress the expression of FLC, a famous genes in flowering time control. For this class, this synthetic block, we did an even broader analysis. We found this block is widely conserved in almost all the Untitled plants. This is a quite interesting discovery, since normally the genes is not so conserved, so broadly conserved. Here we can see this is the basil, angiosperm, the grape, and the soybean Arabidopsis. We can see the red line connects the VRN1 genes. This is the VRN1 gene in Arabidopsis. And we can see this is conserved in almost all the Untitled plants. Indicate this VRN1 gene class is important for all those Untitled plants, and also indicate. And also we found that this gene class is greatly expanded only in Sabindaceae species, in longan, the lychee, and the yellow haw. For the expression, this is quite different from the SVP gene cluster. We can see the expression for all those genes is quite different, looks quite different, not unique, don't have a kind of a similar pattern. So we believe, just like after such a big expansion duplication, many of those genes has kind of been subjected to newer functionalization or sub-functionalization. It has a newer function or has different functions. Yeah, for this class, we also want to do some further functional analysis. The last piece of the last case I want to mention here is a pair of CO-like genes. This is the gene I think is the mostly interesting gene in our study. We combined all the results together, the GWAS analysis, since we don't have a big population, the positive selection, and all the flowering related genes and the differential expression alleles, we found only one gene. This gene is the COL307, and the genes here from the phylogenetic relationship analysis, we can see it is quite close to the CO genes in Arabidopsis. Yeah, so CO genes is famous. It mediates the circadian clock and the control of flowering in Arabidopsis. It is here that controls the expression of FT and the SOC1 expression. So this is important. And for this gene, we found another copy beside close to the 307 one. Here, there is another similar gene, homologous. And more interesting, we found a deletion close to the 307, this gene. And this deletion is heterozygous in the genomes. In the physical genome, we can see it is heterozygous. It is present in one homologous chromosomes, but is absent in the other. But for the other two populations, for the extremely early ones, both chromosomes don't have the deletion. Both of them have the deletion, but for the later maturing cultivars, both chromosomes don't have the deletion, right? It's absent. So, how does this just like a deletion come from? Yeah, based on the sequence analysis, it is similar to the retro transposon. It's a transposon, and it can produce 24 nucleotide small RNAs, which is related to the DNA methylation. So we believe this deletion will have kind of function to regulate the expression of these CO-like genes, and maybe through the DNA methylation pathway. Based on the deletion, we designed a DNA marker of 3 primers. The location of 3 primers is here. Use this DNA marker. We can run a simple PCR to distinguish the 3 populations. One is we can see for the extremely early ones, we can only see a small band, but for the early maturing ones, we have 2 bands, but for the later ones, we have a big band. So we can see this DNA marker is very useful for us to distinguish those 3 populations. Right now, we are using it for our kind of following downstream analysis. So all the results, I just presented, already published just in the first issue of Nature Genetics of this year. You are welcome to read our paper. If you have any questions, just shoot me an email. I'm happy to answer all your questions. To give a summary, recently there is a highlight paper. There is a spotlight in molecular plant, highlighted our results. I think this is a short article which give a really good summary of our story. First, in our study, we obtained a very high quality reference genome of Rui Xia, and also we faced the 2 haplotype genomes, which is quite novel. And we use those haplotype genomes to dissect the origin and domestication history of lychee. We proposed 1 original center and 2 separate domestication events. And also we found a few just important genes for the flowering time regulation, include the SVP genes and the CO-like genes, and also the VRN1 gene cluster. Right now, following downstream functional analysis, we are still doing, So for this part, I would like to thank, this is a really big project, collaborative project. We have around 10, just like institute or universities joined in this project. So there are many people I would like to thank to. Many of them are from maybe abroad, not in China. So we work together a few years. It's a really hard but happy experience. Thank all of them. At last, at the end of this talk, I would like to take maybe 2 minutes to introduce a tool we developed for the genomics analysis and also maybe some other bioinformatics analysis. This tool is, we named it as TV tools. We published 2020 in Molecular Plant in August. After maybe 1.5 years, we got over 1,000 citations. So indicated that many people are using our tools and did many very good analysis and did many interesting studies. And for this tool, I think there are a few advantages over the maybe other tools. I think one is it's simple. It is developed for the web-based biologist. It has a simple iOS logic, just like you just have to set up the input, the output, and click the start, you will get the results. And then also we have a user-friendly interface. You don't have to use command line, you don't have to code, you only have to use your mouse to point and click. And also this is a tool, just like a combo tool with over 150 functions combined together. You can draw figures, you can do basal data analysis, you can do sequence extraction. And also for this tool, it has a very good interactive ability. You can change all the elements easily from the figures made by this tool. Most important, it is free. You can get it easily and install it. Here, I use a few of those plots to show you the interactive abilities of this tool. You can change the color, the font, you can rotation, you can do kind of movement, you can combine those panels together. So this is, I think, is very useful for, especially for those web-based researchers. So if you want to try this tool, just like where to find it, you can easily get it from, maybe you can search it from Google, you can get it from the GitHub, and also we have a group in the Telegram. If you have problems, you can post, you can file a post there. We will answer your questions. Also, you can, if you want to, you can email me, you can email us. We will answer your questions, help you. Just like, I think this is a really useful tool. You are welcome to try it. All right, that's the last slide. I would like to thank you for your time and for all the audience. And I'm a pleasure to take any questions if you have. Thanks. So
great thanks for Dr. Xia for your very fabulous introduction of your recent work. So let's see what kind of questions we have from our audience. Question 1. Any plans to do pangenome of lychee to introduce some other wild species or cultivars from places outside of China, say Thailand, Australia, Africa, etc., so can understand the core genome and non-core genome more comprehensively?
Yeah, this is a really good question. Actually, we have the plan right now. We are doing the pangenome for the lychee right now. But also we have a plan to do more, maybe the Sabbacean species, since as I mentioned, maybe in my second slide, there are many interesting plants in this family, but we will do the pangenome for lychee first, and then I will do some others like long game, maybe combine them together to do some comparative analysis. Yeah, that's a good point. We have the plan. We are going to dissect the core genome, the core genes of maybe the lychee or maybe even the Sabbacean species. But yeah, right now we have the plan, but we need to do it gradually, and we need money and we need people to do that. Yeah, thanks.
Thanks. Question 2. Is there any interaction of the two haplotypes of Fizxiao?
Interaction, you mean just like chromosome level, just like
I guess it's chromosome level.
Yeah, that's an interesting question, but honestly, we didn't check it. I think maybe there are, but we have the data. We didn't do the analysis. I don't know right now. I'm not sure, but I feel it will be interesting. I'm curious just how strong the interaction between those two maybe haplotypes. But right now we don't know. We didn't do the analysis. Yeah, maybe we will try it since we already have the data. It was a trying. Yeah.
Thank you. Question 3. is there any possibility that the subgenome asymmetry is of greater importance than we expect?
我在网上找到这些内容。
Sorry.
So I'm not sure what does the subgenome mean. Just a subgenome, you mean maybe the haplotype or
I guess
yeah.
it's two haplotypes. There are
yeah, maybe the structural variation, or maybe or even the expression different expression of the subgenomes, just like the expression dominance, something like that. Yeah. I would say yes, just like I think the subgenome asymmetry, just like the imbalance, right? Just like the imbalance between the two haplotype genomes, just like the expression or maybe the structural variations. I think we already give a good example, just like the deletion related to the close to the CO genes. We believe it is related to the flowering time control. So this is, I think, one haplotype genome has a deletion, the other one doesn't have. So this is, I think, is one point. And the other one, since for the I think the expression level at the expression side, I think there are some maybe expression bias. As I show, we have identified 13 maybe 1,000 differential expressed alleles. That means those alleles has different expression. So I think it will have kind of function to the features, just like I think it's even related to the heterosis, right? Just like heterosis, just like I think that's why, since in our story, we even believe since why the physics shell is popular in China, because the fruit is of good quality, and it comes from the hybridization between maybe the early one, the Yunnan wild, and the late ones. Yeah, so I think maybe the combined together of those two genomes maybe have some effect on the quality of the fruit, made the fruit better. So that's why physics shell is more popular right now. Yeah.
Great, thank you.
Mm-hmm.
Next question. What kind of methods do you use when you try to identify the DEAs?
Oh, what kind of method? You mean just like, yeah. I think maybe in our first round of revision, one of the reviewers asked this question for the identification of DEA, the differential expressed alleles. Since we have two sets of haplotype genome and two sets of genes, we just combine them together, merge them together. So we will map our RNA-seq data to the merged genome. So only choose the best hit rate to calculate the expression value, and then we will compare the expression between alleles, just like if the fold change is bigger than 2 and the FPKM is bigger than 1, and also this kind of differential expression happens in at least 2 libraries. We regard it as a differential as a DEA. So we have a couple of criteria in our identification. So if you are interested, I think maybe in one of the supplementary plots, we list all those standards we used for the screen. Yeah.
Okay, great, thank you. Another question. When you try to phase the assembly of the 2 haplotypes, do you meet any problems?
Yeah, this I think one of the kind of novelty of our story is we separated the genome into 2 haplotype genome. We found some we got problems. Yeah, even in the first revision, one of the reviewers questioned our results. That's why in the revision we added the 10 genomics data, just like for more accurate calling of haplotypic SNPs. So I think yeah, we do got problems. One is we tried all the available tools, include OHIC and some other tools. We did the we compared them, but most of them doesn't work very well in our case. And also we tried a different kind of flow, just like workflow. We at first, I think the first try we did the phasing after the assembly by canoe. After the canoe assembly, we try to phase the contiguous. But at that stage, I think the canoe is not a good tool, since it will merge the kind of, it will cause a chimeric assembly. So if you assemble the reads into the contig, and then you cannot separate them after that. So in our revision, we just used a method just to phase the original seed, the original reads of the pack bar reads at the very beginning. After phasing the reads, we do the assembly after that. So we got even better results. I think this is the method we used in our phasing, but I would say maybe different species is different. Yeah, just like since the plant we use is physics, which has a really high heterozygous rate, is over 2 points, right? Many of the plants doesn't have such a high heterozygous rate. Maybe I think you need to test different method or maybe algorithm to do the assembly. Yeah, that's my suggestion. Thanks.
Thank you very much for such detailed answer. Another question. May I know, do you have any future plans about the circadian clock studies of your focus genes?
I think you are talking about the CO-like gene, right?
Yeah.
Yeah. We really want to do some work on this part, and we are trying to establish even the transformations gene transformation system in Lychee, but it is difficult right now. We don't have a mature system for it, but we are trying to we are trying to establish maybe a model system, even use some other subadation plants, as I think right now we just like here. Can you still see my screen right now? Yeah, just like I think there are some species is different, just like here, even there are some wild plants which can flower easily, but for lychee and longan, it takes a few years to flower. So it's really difficult to test the circadian and the flowering time control. So, but we are working on it, but it is difficult. Yeah.
Great, thank you. Due to limited time, unfortunately, we cannot answer more of the questions. We will get back to you through email after the webinar. So since time is up, so if there is no other questions from our audience, we will end today's webinar.
Okay, thank you. Yeah, I'm happy to take any questions if you have, and you can maybe just email me. Yeah. All right.
Okay, great. Thank you, Dr. Xia, and thank you all for our audience.
Okay, thank you. Thank you for your host.
Thank you. See you again. Bye-bye.
Bye.
Bye-bye.